Hi, I'm Kieran. I'll be talking to you about my PhD project so far, which has been on targeting neutrophil cell death as a treatment for multimorbidity. So first of all, why am I even looking at multimorbidity? So multimorbidity is defined as the presence of two or more chronic conditions in any one person at any one time. And in 2015, around 54% of all people aged 65 or over in the UK exhibit some kind of multimorbidity. And this is a figure which is expected to rise by 2035. So one of the biggest, most prevalent forms of multimorbidity are those which involve chronic inflammatory diseases, and that's what my research is focusing on. So chronic inflammatory diseases, such as COPD and atherosclerosis, account for around 70% of all deaths worldwide. And despite this massive prevalence, current treatments for these disorders, like anti-inflammatory drugs, have had very limited success in resolving chronic inflammation. So that shows a massive need for new treatments which can treat these diseases. And one strategy to do this is by targeting neutrophils. So neutrophils are a key component of the innate immune system and are the first responders to any cellular damage or invasion, like through a bacterial pathogen. And neutrophils are able to remove harmful pathogens in three ways. So first of all, we have here on A, that's phagocytosis, where the neutrophils are able to engulf bacteria, and that can remove them very safely from the body, and they're digested by degradative enzymes and things like that. Another method is degranulation, shown there by C, and that's when they release all these degradative materials into the extracellular space to kill the bacteria. But this also causes damage to the host tissues, so that's pro-inflammatory. And as well as this, neutrophils have a specialized form of cell death shown in the middle there, which is NETosis. This is where neutrophils can decondense all of their nuclear material, and when it gets covered in all these degradative enzymes and reactive oxygen species, it's been released into the extracellular space to make a net to trap and kill pathogens. But again, this is very pro-inflammatory and can cause a lot of host tissue damage. So realistically, we don't want them to do them too, we want them to phagocytose and die nicely. So that brings me to my hypothesis for my project, which is that the EGFR/ERB inhibitor Neratinib and the protein kinase C beta-2 inhibitor Ruboxistaurin are able to target neutrophil cell death in order to resolve chronic inflammation. And to test this, I have an overall aim, which is to determine whether Neratinib and Ruboxistaurin are effective treatments for multimorbidity. And I've split that into 4 sub-aims, because that would be ridiculous to test. But first of all, today, I'm just going to be talking about, well, that should be on the first one, which is to explore the anti-inflammatory mechanism of action of Neratinib and Ruboxistaurin in healthy human neutrophils in vitro. So first of all, I'll talk about NETosis, which is the first neutrophil cell death mechanism, which I've already talked about briefly. So in order to test whether my drug is able to inhibit NETosis, as it's pro-inflammatory, want to inhibit this process, first of all, I have to take blood and then process this blood to be just neutrophils. I'm then able to treat these neutrophils with Ruboxistaurin and then stimulate NETosis using a well-known inducer known as PMA. I'm then able to stain these nets and the DNA released in order to see how much DNA is released by these cells. And you can see here I've got kind of a cartoon of a net, and then these are a picture of actual nets under the microscope, which I've got. So using this method, I've been able to show that the protein kinase C beta-2 inhibitor Ruboxistaurin is able to significantly inhibit net formation in vitro. So as you can see on the top here, we've got unstimulated neutrophils, and you see the nice tight little balls of cell, basically. And then with PMA, they all just kind of explode and spit out all of this material in DNA. And then with the drug, although some do still react to the PMA, there's a massive reduction. You can see most of the cells are still intact down there. So that's what we want to happen. We want to reduce the release of this stuff so it doesn't damage the host tissues, basically. So as well as NETosis, neutrophils can also die by apoptosis, which is very preferable because these cells then die into tight little balls, which are then removed by macrophages, so they're cleanly removed from the cellular space, so no host damage. So I've shown that I can inhibit NETosis, and now I want to induce apoptosis. So to do this, I do exactly the same as I did before, isolate the neutrophils from healthy human blood, and then I treat them with Neratinib. I'm then able to move the cells from the wells onto slides by centrifugation, so that splats the cells onto the slide, and then I'm able to use light microscopy to look under the microscope and quantify apoptosis. So you can see I've got a very nice, healthy neutrophil there, and then the apoptotic neutrophil is a little dark ball because it's starting to die, basically. So then using light microscopy, I can quantify the amount of dead and live cells in each sample. So then using this, I've been able to find that my ERB inhibitor Neratinib is able to significantly induce neutrophil cell death in vitro. And this is dose dose responsive, basically. So as the dose increases, it does kill more neutrophils. And this is a very clinically relevant concentration which I've been using, which has been used for cancers in the past. So this is very translatable to future work. So in summary, so far I've been able to show that Ruboxistaurin is able to significantly reduce the amount of nets produced by PMA-stimulated neutrophils in vitro. And I've also been able to show that Neratinib is able to significantly induce neutrophil cell death in vitro. And finally, I'm going to talk a little bit about my future work because it's a massive part of my project and I've started working on it now. So that's the mouse models. So first of all, I'll be using a mouse model of COPD to determine whether these treatments are going to be successful in reducing the lung damage seen in COPD. And that's in particular the alveoli damage. And then as well as this, I'm going to use a mouse model of atherosclerosis to determine whether these treatments are successful in reducing net levels observed in atherosclerotic plaques. And finally, I'm going to use a combined model where one mouse has both diseases to see if these treatments still work in the case of multimorbidity. So finally, I'd like to acknowledge people who've helped me on my project so far. So it's my supervisory team and the Healthy Lifespan Institute and the Dunhill Medical Trust.
Thank you very much, Kieran. So do we have any questions?
That was really lovely. I just wondered, you've looked at NETosis and apoptosis. Do your drugs target phagocytosis as well? Because if you're changing that, then you could be changing the function of the neutrophil completely.
Yes, so the picture which I showed there is from one of my atherosclerosis assays. So that's specifically the eating of dead neutrophils. So I've been able to show that Neratinib does induce neutrophil cell death. And one of our papers using Neratinib found that in mouse models there was an increase in atherosclerosis. But we couldn't tell if that was because there was more dead neutrophils or if it was actually doing something. So I've been doing a lot of atherosclerosis assays, and the preliminary data looks like my drug is enhancing atherosclerosis rates, but that's preliminary data at the minute, but it is looking that way.
Thank you very much. We have a question here.
Thank you, that's really fascinating. I was just wondering if you know what influences the response of the neutrophils down one of these three paths that you mentioned, and if that's something that changes with aging.
Yeah, so aging is something that does contribute to NETosis as well. So when you get older, you run out of bone marrow, and then your neutrophils are sort of a bit more weary than in younger people. So aged neutrophils are more primed to NETos than younger neutrophils, and that's consistent in older patients as well as younger patients, but they're more ready to produce nets. So as well as using this in disease states such as COPD, we think that it could be very good for people with arthritis, for example. So Ruboxistaurin was originally used for arthritis, and they think that could be that in older people, nets are being made in these joints, and using Ruboxistaurin could reduce the nets and hopefully reduce the age-related diseases as well.
Thank you. Any more questions? We've got a couple. One at the back there.
Yeah, this might be a bit of a silly question as someone who doesn't work on neutrophils, but how is it that PMA stimulates NETosis, and is it possible that your drug is targeting that stimulation rather than the nets?
Yes, so that's a very good question. So basically, Ruboxistaurin is a protein kinase C inhibitor, and PMA is a protein kinase C activator. So it makes sense that my inhibitor was stopping this process, but that's a very well characterized way to make nets. But on this slide as well, you can see that I've got another condition, which is CIA. So that's a stimulator which does work through a different method of creating nets, so it doesn't even interact with a protein kinase C pathway. But Ruboxistaurin is still almost exactly the same as in PMA-reduced that. So we think that Ruboxistaurin is sort of inhibiting all NETosis, even the ones which are specifically targeting Ruboxistaurin's pathway.
Cool.
Thank you very much. I think we have one question here, and that will be the last question.
Hiya, lovely talk. I might have missed, but are you planning on using them together, or is it just that you were...
So at the minute, we're going to use the drugs independently of each other, but we're sort of thinking at the minute that one's going to be better at one disease and one's going to be better at the other disease, and we're going to hopefully see if they can still work independently and then maybe combine them, but we're having a little bit of trouble with the multimorbidity model, so we're not too worried about treatment strategy just yet, but...
And I was also wondering if, as like a follow-up, if you think using a PKC inhibitor, if you would expect off-target effects, because obviously PKCs are involved in quite a lot of stuff.
Yeah, so because Ruboxistaurin has passed phase 3 trials, there doesn't seem to be too many negative side effects. I think the main one was sort of a bit of diarrhea and intestinal issues, but that's still very mild, and with the concentrations we're using at, it's slightly lower than it was used in that trial as well, so we're hoping that there's not going to be too many negative effects of using them, but we're not 100% sure at the minute.
Okay, thank you.
Thank you very much, and thank you very much, Kieran.